The topic of today at Medical Sciences, we want to discuss about the structure of triglyceride. Others call it triacylglycer. So we want to see its structure, the properties, and then we shall end by looking at the properties or chemical properties of fats, and then the characterization of fats. And if you are new on this page, please go on the subscription button, subscribe, like the video, and share to many others so that they can continue to learn with us. So triglycerides, which we can call triacylglycer, this is an ester. It is an ester of hydrocarbon chain. It is an ester of a hydrocarbon chain, and this hydrocarbon chain, it has the carboxyl group, the carboxylic group on the terminal. So meaning triacylglycer, they are made up of the brief summary of it is RCOOH, whereby R is indicating the hydrocarbon chain, and this R group, which is indicating hydrocarbon chain, it is hydrophobic. This is hydrophobic chain, whereas the carboxyl group is hydrophilic. The carboxyl group is hydrophilic. And that's why we end up by saying that the triglyceride, they are amphipathic. They are amphipathic. Why do we say it is amphipathic? Because it contains both the hydrophobic chain, which is a hydrocarbon chain, together with the carboxyl group, which is hydrophilic. So it has the polar and the non-polar part, and that's why we say it is amphipathic in nature. Another thing we have to know about this triglyceride, apart from being amphipathic, it is insoluble in water. It is insoluble in water. That is another thing we can talk about these triglycerides. So its structure, it contains glycerol. It has, if you get glycerol, and this glycerol, and combine it with the carbon chain, whereby we are going to combine it with the fatty acid, 3 of them, we are able to form triacylglycer. That's what. If I have glycerol, glycerol is a 3-carbon sugar containing of CH2, CH, and CH2. And these ones, all of them end with the hydroxyl group. This has a hydroxyl group, and this has the hydroxyl group. This is carbon number 1, carbon number 2, and carbon number 3. So this is the structure of glycer. So if I come and I add 3 fatty acids, and what are these 3 fatty acids? We are having fatty acid 1 with, so this one, you can make it face this side. I'm representing it in this form. Then we have the second fatty acid chain with the OH group. Then we have the other third with the OH group. So if we add 3 fatty acids, this is the fatty acid number 1, number 2, number 3. and this R is showing a variable hydrocarbon chain. So when we react them together, we are able to form triglyceride. How is that? The hydrogen group on the glyceride reacts with the hydroxyl group on the fatty acid, resulting into formation of water. Even this one, the hydrogen group on the glycerol, and the hydroxyl group on the glycerol combines with the hydroxyl group on another second fatty acid, form this water. And even this hydrogen group on the glycerol combines with the hydroxyl group, this forming another water. So the end result, we are forming 3 water molecules. Here there was another formation of water, and this is how the ester bond is formed. But now we are going to have CH2, then we are remaining with this oxygen, and this oxygen has been bonded to this carbon. And this one becomes 1. Then we have the second one with the oxygen and another carbonyl group forming the R2. Then we have the third one. So if we have only one fatty acid chain, this one we call it monoacyl glycerol. If it is having 2, we call it diacyl glycerol, that is DAG. And when it has 3, we call it triacyl glycerol. So this is the structure of triacyl glycerol. So during hydrolysis, if I break the bond, the fatty acid at carbon number 3, I form diacyl glycerol. If I break the second bond and I remain with the one fatty acid, I form monoacyl glycerol. So this is the structure showing how triglyceride or triacyl glycerol is formed, and this is the ester bond formed. So the ester bond is this one formed in between these. So this is what we regard as the ester bond. So we are indicating that this is the ester bond. So in triglyceride, the type of bonding is known as ester bond. So hydrolysis enzymes which break down triglycerides, like phospholipase, like lipase, they come to break the ester bond by adding water molecule. And among the properties or the functions, because we saw the properties, it is amphipathic and insoluble in water. And this triacyl glycerol, its functions, its major function in the body, it is the major energy fuel. It is the energy deposit, whereby energy or fuel is kept in form of triglycerides within the adipose tissue. So in case all carbohydrates are used up, we can now use the energy from adipose tissue. And one triglyceride molecule gives 9 kilojoules of kilocalories of energy. One triglyceride. Or we used to say it can bring about 129 ATPs, 1 molecule of triglyceride. And apart from being the energy storage form, also triglyceride can be used as a precursor for formation of phospholipids. You can use it as a precursor for formation of phospholipids in the body, which act as membrane fats. So these are some of the major 2 functions of triglyceride, which we have seen its structure, this illustration. So after looking at this, now we are going to look at the chemical properties of fats, or you can stroke them some triglycerides, or what we can call fats in general, or oils. And one of the property is hydrogenation. And what is hydrogenation? Hydrogenation is addition of hydrogen, and this hydrogen is being added to unsaturated fatty acids. So addition of hydrogen on unsaturated fatty acids in the presence of nickel catalyst. Leads to hardening. This one results into hardening of fats. And this hydrogenation process is being used in industries or commercially in industries to convert liquid oils from plants to solid. For example, we can get oils from plants and we harden them into solid form, and that is where we can form margarines, or with ghee. Plant ghee is got by hydrogenation, whereby they are hardened so that they can be sold in a solid form. For example, we have seen margarine, even plant ghee, plant oil ghee are manufactured by hydrogenation, whereby hydrogenation bring about the hardening effect, and it occurs in the presence of a catalyst known as nickel catalyst. So that is the first chemical property or reaction of fats. Then the second one is what we know as rancidity. And what is rancidity? Rancidity is that as the fats are aging, when fats are aging and they are exposed either to light or air or heat, they tend to develop, or even exposed to the lipases, enzymes like lipases which hydrolyze, they tend to form bad odor. They develop bad odor, what we call smell, and bad taste. So rancidity is the development of smell, bad odor, and taste of these fats or oils upon aging when they have taken long. And this one we are seeing it is either due to exposure to light or air or enzymes like lipases. They bring about this. And this development of bad odor and taste in fats, which we are calling rancidity, it can be categorized as oxidative rancidity. It can be hydrolytic rancidity, and sometimes we have also microbial rancidity. So these are some of the forms of rancidity, whereby oxidative rancidity, it is brought about by various oxidative processes. That these oxidative processes, they bring about formation of peroxides. And these peroxides can continue to undergo decomposition. They can decompose to form aldehydes. And these aldehydes can even lead to formation of toxic substances in fats, which can bring cancerous cells, or can lead to which are carcinogenic. That is what we can see under oxidative. And it is majorly due to exposure either of fats to light or heat, or when it is exposed to air. They can cause that formation of peroxides. And these peroxides, when decompose, they form aldehydes, which can be carcinogenic in our body. Then under hydrolytic rancidity, This is due to action of enzymes like lipases, whereby these lipases break down the ester bonds in fats by addition of water, leading to the development of body odor and taste. That is what we can see. Then under microbial, we see even microorganisms like bacteria, microorganisms like molds or fungi, using their enzyme lipases to break down the fats. And this one results into body odor and taste of the fats. And how do we preserve fats from aging? Due to these factors. To prevent body odor and the taste of fats, we can use preservatives like antioxidants. For microbial, we can use sterilization methods. Sterilization methods can help us to rule out microbial rancidity. Whereas other forms, we can easily use antioxidants. Another form of preserving is using antioxidants. Antioxidants include vitamin E and vitamin C. Ascorbic acid and tocopherol can be one of the antioxidants which are used to preserve fats from undergoing rancidity. So if you want to prevent development of bad odor and bad taste of the fats, you can use antioxidants like tocopherol, that is vitamin E, or you can use ascorbic acid, which is vitamin C. Then for microbial, you make sure they are well sterilized to prevent this rancidity. That is what we can look at. Then the last reaction of fats, and this one especially occurs on triglycerides, is saponification, whereby triglycerides in the presence of alkalis like sodium hydroxide or potassium hydroxide, they are able to form glycerol. They form glycerol and a salt of fatty acid. And this sort of fatty acid is known as soap. So the reaction whereby fatty acids like triacylglycerol react with alkalis, forming glycerol and salt of the fatty acid, which we know as soap. So in case I have a triglyceride, if I have a triglyceride, this is the triglyceride with an ester bond. So this triglyceride can, when it reacts with alkalis like sodium hydroxide, it undergoes saponification. Producing glycerol, forming glycerol and a salt of fatty acid. And we are going to form 3 salts of fatty acid, whereby we shall form the salts of, so here the hydroxyl group on this, the oxygen group here, we come and add the sodium. Then this one donates the hydrogen and sodium is added at this point. And this is what we call soap. This is known as soap. So this is how soap can be formed in industry. And we shall see how can we determine the ability of the fatty acid to form soap using saponification number. So this is how we are saying in case we have triglyceride, it reacts with alkali like sodium hydroxide or potassium hydroxide to form glycerol. and soap. And this is how it is formed. This is what we know as saponification, which we are using commercially to form soap for sale or for using as a detergent. So lastly, we are going to look at characterization of fats. And under characterization of fats, we are going to look at 3 components. And the 3 components we are going to discuss with you under characterization of fats. They can be characterized by saponification number, they can be characterized by acidic number, and lastly by what we call, so there is saponification number, acidic number, and then we can also use iodine number. And under saponification number, what is it? This is the amount, or you can say number of grams of potassium hydroxide or sodium hydroxide, which is required to saponify 1 gram of a given fat. So the number of grams of potassium hydroxide or sodium hydroxide which is required to saponify 1 gram of a given fat is what we call as saponification number. And saponification number is inversely proportional to molecular weight. Of fats. What do we mean? We are saying if it is inversely proportional, it means the shorter the chain, we are going to see that short chain fats, fatty acids or fats, they have higher saponification number. So the shorter the triglyceride, the higher the saponification number. So meaning short fats, they are able to form soap easily compared to long chain triglyceride. That is what saponification number we can use when we are going to produce soap in industries to know which fats can give us soap easily compared to others. So we have seen short chain gives soap easily compared to longer fatty acid triglycerides. Then the next we can talk about is acidic number. And acidic number, this is the number of grams of potassium hydroxide which are required to neutralize or to saturate, but majorly the best word is to neutralize free fatty acids, to neutralize 1 gram. Neutralize 1 gram of free fatty acids. So the amount or the number of potassium hydroxide required to neutralize 1 gram of free fatty acids is what we call acidic number or acidity number. And this one is used to determine the edibility, to see if fats are edible or not edible. And we know that fats with free the free fatty acids are not good for health. So if the acidic number is high, the acidic number of the fat is high, it means that this fat is not good for consumption. Not good for consumption. So we can use the acidic number or acidity number or the acid number. We can even call it acid number. We can use the acid number to determine how fit is the fat for consumption by human being. And we find that fats which have high acid number are not good for consumption, and fats with lower acidic number, they are good for consumption because they determine the number of free fatty acids present in these lipids. So that is the application of acid number. Then lastly, we are going to discuss with you iodine number, whereby iodine number, the iodine number here, it is the number or the amount of iodine which is required to saturate the number of iodine required to saturate 100 grams of unsaturated fatty acid. If I have an unsaturated fatty acid, we want to see how many number of iodine molecules are needed to saturate 100 grams of a given unsaturated fatty acid. And the higher the iodine number, so the significance of iodine number is to determine the degree of saturation. The degree of saturation of fatty acids. And because we know that iodine prefer to attach on the double bonds, this iodine prefer attaching on the double bonds of fatty acids. So the higher the iodine number, we are going to end up by saying the higher the iodine number, the more this fatty acid is saturated, the more the double bonds, or the more the fatty acid is unsaturated. So we are using the iodine number to distinguish between the saturated and the unsaturated fatty acids, whereby we are going to see that unsaturated fatty acids have higher iodine number compared to the saturated acids. So this is what we can discuss with you in this video, whereby we have looked at the structure of triglyceride. Please, as a student of biochemistry, you need to know how to draw the structure of triglyceride and how the ester bond is formed during reaction of glycerol with 3 free fatty acids. And we have seen that we can hydrolyze triglyceride to form diacyl glycerol and monoacyl glycerol. Then we have gone ahead to look at the function. Then we have looked at the chemical properties. Don't forget hydrogenation, which is used commercially to harden liquid oils from plant products to form solid products by adding hydrogen on the double bond. Then rancidity, which is aging of fatty acids, it is prevented by adding antioxidants like tocopherol, vitamin E, and ascorbic acid, which is vitamin C. Then we have looked at saponification, how commercially we can prepare soap using fats in the presence of alkalis, forming glycerol and salt of fatty acid, which is soap. And we have seen we can determine the ability of the fat to be saponifiable by determining saponification number. And whereby the higher the saponification number, meaning the fat can be able to form soap. Then we have looked at characterization, whereby you have looked at saponification number, acid number, and iodine number. Thank you so much for learning with us at Medical Sciences. Please remember to subscribe if you haven't. That is the only gift you can give us to say thank you. And remember to like the video so that you can continue to follow with us. Thank you so much.